And this was the question that intrigued Beth Stevens most. If microglia, which researchers now understood, were akin to the white blood cells of the brain, and they were altering brain circuitry, perhaps microglia didn't always get it right. What if, instead of just pruning away damaged or old neurons, microglia were sometimes mistakenly engulfing and destroying healthy brain synapses too? Increasingly, disorders of the brain, from depression to learning disorders, were being understood as circuitry disorders. Certain synaptic connections in the brain were not firing and wiring together as they should. Beth explains, what if these little overlooked glial cells we were setting out to study were at the center of it all? Her hands spread wide with excitement. You can imagine how, if you have too much pruning or too little pruning, things go awry. You could have too many synapses or not enough synaptic connectivity. And you can imagine, given how our brain works, if that connectivity is even slightly off, that could potentially underlie a range of neuropsychiatric and neurodevelopmental disorders, as well as cognitive impairment. Could it be that sometimes microglia were the brain's sculptors and protectors, the angels of the brain, and sometimes they were the brain's untimely assassins? Was it possible that scientists had, for so many decades, missed something so huge? No one knew. If Beth could prove this grand, complex hypothesis, it would completely change our understanding of brain health from womb to grave. But in order to prove her hypothesis, she first had to get a better look at these little cells. Beth hypothesized that if, indeed, microglia were engulfing and destroying synapses, then she would see chewed up synapses inside the microglia. That was the big question, Beth explains. Could we find pieces of synapses inside the microglia? Dory, my postdoc, figured out a really clever way to allow us to see if that was indeed the case. Dory Schaefer, now an assistant professor of neurobiology at the University of Massachusetts Medical School, explains their experiment, one that is now universally considered a landmark study in the field of neuroscience. In order to get a closer look at how microglia were interacting with synapses, Dory injected dye into the eyes of mice. This dye then traced down from the neurons in the eye nerves and deep into the brain. This, says Dory, made the brain's synapses glow a bright fluorescent red. Microglia, on the other hand, were stained fluorescent green. And this, Dory says, allowed us to clearly see both the synapses and the microglia, since each glowed a distinct fluorescent color. This process, simply finding a way to accurately see the interplay of synapses and microglia inside the brain, took nearly a year. Dory recalls, one weekend I was in the lab by myself, imaging and re-imaging microglia and synapses. I'd looked through the microscope a million times. Then suddenly I saw these little red structures, synapses glowing like fluorescent red lit up dots. And these red dots were inside the belly of the green microglia. Dory was stunned. I kept thinking, we were right. Microglia are eating synapses. I was staring at proof positive with my own eyes. The synapses were inside the microglia, and microglia were chomping them to pieces. Beth recalls Dory running into her office that Monday morning with photo images in her hand. They're in there, Dory told Beth. The synapses are really inside the microglia. We can see it. It was such a high-five moment, Beth recalls. Microglia were like tiny little Pac-Men in the brain, and brain synapses were in the bellies of the Pac-Men. We were on to something really wonderful, really novel. This was deeply important in terms of looking ahead to microglia's role in disease. Beth and Dory rolled up their sleeves. We were both really fired up, but it was also really hard and stressful, Beth recalls. She was pregnant with her daughter Zoe and had a toddler at home. Dory and I knew we had to crank. We had so much adrenaline, but we also wanted to be rigorous and take our time doing our control experiments and data analysis. We wanted to do it right and publish it before anyone else. It was a tall order. The following weeks and months were reminiscent of Beth's early days as a scientist, back when she'd started at NIH. Many nights she'd look up at the clock and it would be close to midnight. It just wouldn't make sense to go home, she says. But one thing had changed compared to her early days as a lab tech. Beth no longer had to sleep on a pile of clothes under the lab conference table. Laughing, she tells me, Dory gave me a blow-up mattress as a present. I put it under my desk, and if it was really late, I'd crash there. In 2001, Beth and Dory submitted a paper outlining their findings for publication and review. Beth had recently given birth to her second daughter, Zoe, and now had 2 young children at home. And Dory had recently gotten married. It was a watershed year. In 2012, their seminal paper was published in the journal Neuron. It was the first scientific study offering evidence that microglia were pruning and eating away synapses. Not surprisingly, the scientific world exploded with the news. Their study was later cited as the journal's most influential paper that year. Meanwhile, researchers at the European Molecular Biology Laboratory in Italy showed that microglia could be especially overactive in the brain's hippocampus, an area crucial to mood and memory. By engulfing and removing healthy synapses in the hippocampus, microglia were causing a loss of brain matter in a part of the brain that was known to be deeply affected in depression, anxiety disorders, obsessive compulsive disorder, and Alzheimer's disease. In these diseases, the brain's hippocampus had been shown through PET scan imagery to be visibly atrophied. These revelations solved a decades-old mystery. In many different neuropsychiatric and neurodegenerative diseases of the brain, healthy synapses disappeared, but no one had ever understood why. Suddenly, it all made complete sense. Even though microglia were trying to protect the brain and make it healthy and strong, just like white blood cells in the body, when microglia detected that something was off, an overinflux of stress hormones, an infiltrating virus, chemical, or pathogen on the scene, they all too often went too far, removing every synapse hanging out in the neighborhood. And that understanding that this little boring cell that had been hiding in plain sight was an immensely active and sometimes hyperactive immune cell changed everything.